various repetitive elements that that will lead to ambiguities in the assembly process and will prevent us from getting a continuous genome sequence at the end. And bacterial genomes contain several types of repeats. We have short tandem repeats, longer tandem repeats. We have interspersed repeats that occur in different positions in the genome. We have integrated bacteriophages. We have the ribosomal RNA genes, of which there are several in a typical bacterial genome and placed in various positions. And all these repeats will prevent us from getting to a fully circular genome in the end. And this figure tries to explain why repeats cause problems. It's maybe a bit complicated, but if you look at the top here, we have a sequence which goes from a green unique region here into 3 red repeats. So these repeats are identical. This repeat here is identical or more or less identical to this one, which is more or less identical to this. And then we have unique sequence again in the other end. And again, at the top here, it shows us how an assembly ought to work. We should be, from all these fragments we have here, be able to assemble the sequence we have up here with these 3 repeats flanked by unique sequences. But in real life, this will not happen because this fragment here will be more or less identical, at least in the end, to this fragment here and so on. So instead of getting the assembly of 3 separate repeats here, we will end up with something indicated down here, where all these red sequences from the repeat region will assemble into 1 or maybe 2 instead of 3 repeats here. And this will also lead to gaps in the assembly because this region here cannot easily be assembled with the region here. So instead of getting 1 circular complete genome sequence, we will get a number of fragments, so-called contigs. And we will, at least in the first round, not really know how these contigs are related to each other, which one are the neighbor of which ones and so on. We can, if we look at how the fragments map to the sequence or how they are arranged here, we can see that strange things have happened because this region here will have a much higher coverage than the unique regions in the end here. So we can understand that something has gone wrong because we have a much higher fragment density here than we have here. But we can't really get easily from the assembly we have down here to the correct assembly up here. The easiest way to solve this problem would be to have, instead of short sequence reads like the ones we have here, have sequence reads which are so long that they span the entire repeat region, all 3 repeats, and have this green unique sequence in the ends. Reads like this would make it possible to realize that we actually have 3 repeats in this region instead of 1 or 2 repeats as the assembly would like us to believe. So the solution, at least the simplest solution to the assembly problem, would be to have reads which are so long that they span all the possible repeat regions we have in the genome we are studying. And if we have these long reads, the assembly process will be simpler, and the assembly will actually reflect the real structure of the genome. If you sequence a bacterial genome these days using short read technology, which I will return to in a little while, you will end up, instead of having a nice circular genome like this one, you will have a number of contigs, which all together will cover more or less the entire genome, but will not really know how these ones are arranged relative to each other in the intact genome. But we'll have the complete, or almost the complete genome sequence, but it will not be completely ordered. And that, of course, could be a problem. In many cases, it's not really a problem, because what we are interested in is the complete sequence of the genome. And we have at least more or less the complete sequence of the genome in the assembly of contigs we have here. But in some cases, it certainly would be much better to have an intact genome like the one we have here. And there are examples that genomes which have been known at this level with a number of contigs which are not sort of joined together. Once one manages to join them together and get a circular genome, one will at least sometimes discover facts about the genome that we could not see in this collection of shorter contigs. A small look back here is I have this picture mainly to sort of illustrate the difference between sequencing genomes 25 years ago and sequencing genomes today. So this is the kind of factory that was used to sequence the human genome. And several of these factories of sequencing instruments and robots were spread around the world, and altogether they sort of managed to produce a draft sequence of the human genome in 2001. So this factory consists of several automated DNA sequencing instruments, and in addition, robots for growing bacterial cultures, for purification of DNA and performing the sequencing reactions. In Britain, I've been told that they didn't use robots that much because they preferred instead to hire preferably females from around the factory, females without work, and give them a crash course in setting up sequencing reactions and have them perform them rather than robots. But anyway, in order for a system like this to sequence the human genome, you need a lot of factories, and you need to run sequencing reactions all the time over periods of years. So there was a lot of work involved in achieving this draft human genome in 2001. Note here that each of the instruments could generate, again, 32 to 96, dependent on the instrument, sequences, each of them about 400 nucleotides long, and one could run 3 runs per instrument per 24 hours. So a lot of work to obtain sequence enough to assemble a human genome. But then things happened. New sequencing technologies were developed, and the one I'm illustrating here is the technology used in a company which originally was called Solexa, but which later was bought up by Illumina, which is a big company working with various kinds of instrumentation for genome studies. And the principle behind this technology was you take your genome, you fragment it, preferably using mechanical stress or whatever. You fragment it into fragments of suitable size, And these fragments you bind to a surface. So these long here are fragments which have been bound, and the shorter ones here are simply oligonucleotides, which we use to capture these longer fragments here. And then by a really ingenious technique, each of these bound fragments were amplified, and the amplification using PCR was performed in such a manner that we get these clusters of identical DNA sequences which are located in a specific cluster on this substrate we have here. So for each, these two fragments here have then in the amplification process given rise to about 500 copies of itself, which are bound to the substrate in more or less the same position as the original fragment was bound. And these clusters of DNA could then be sequenced, and in the Illumina technology, they use a technique where they, instead of using normal nucleotides to synthesize DNA, use nucleotides, which I've tried to illustrate here. I'm not sure this figure really shows it clearly for you, but anyway, we have a nucleotide here which has been modified in two ways. One modification is that it has a fluorescent tag in the end, and the color of the tag will be different for the 4 different nucleotides. And in addition, the 3' hydroxyl group of the deoxyribose here has been blocked, so that once a nucleotide like this has been incorporated in a polymerase reaction, the process cannot continue, because we can't add a new nucleotide to this one because the receiving hydroxyl group here has been blocked. Which means that in this process, we incorporate one nucleotide for all these millions of clusters which we have on the substrate here. And we identify which nucleotide has been incorporated in which cluster by looking at the colors. So this shows a small outcut of the substrate here, and for each of these dots here, we have a cluster of identical DNA molecules which have functioned as templates in a polymerase reaction, and we can tell which nucleotide has been incorporated in this step by looking at the color. Once the incorporated nucleotide has been identified, one will attack this system here by using chemicals which will, first of all, remove the fluorescent group so that the molecule no longer is fluorescent, and also remove this blocking group on the 3' hydroxyl group so that we have in the end of the growing DNA chain here a nucleotide that can accept a new nucleotide. So the polymerase reaction can continue, and we can add the next nucleotide, which again can be identified by looking at the digital image of the substrate here. So for each step here, we will get a different pattern of colored dots here, and the pattern will tell us what happens in each of these dots. So in this way, you can sequence millions of DNA fragments at the same time. Although each incorporation here will take time, because we have to incorporate a new nucleotide, we have to identify it, we have to remove the fluorescent group and the blocking group here before we can proceed to the next step. So each of these sequencing steps are quite time consuming, but since it's performed on millions or billions of DNA molecules at the same time, the output of such an instrument can be enormous. So for I've shown here the output from 2 of the Illumina instruments. The next SEC is a big instrument, which will start with 1.2 billion clusters like this one. And all these clusters are then sequenced at the same time. And the read length we can obtain with this instrument is these days 150 base pairs times 2, because once we have read one strand, we can read the complementary strand as well, which also will be present in these clusters. So one instrument like this will per run give us 360 gigabases of DNA sequence, which is vastly more than the previous generation of sequencing instruments could give. Illumina has smaller instruments as well. MySec, which I've used in for a couple of genomes, which has a lot lower number of clusters, 25 million, but still quite impressive. I mean, 25 million sequences, separate sequences at the end of the experiment. MySec can read longer sequences. The instruments that are available today would read 300 base pairs, which we again can multiply with 2 because we can read the complementary strand as well. And the maximum output from each run is roughly 15 gigabases. So within not many years, sequencing technology was completely transforming the way we had to think about sequencing genomes, because we suddenly were in a position where a single instrument from Illumina could give us much more sequence than 10 or 20 of the factories which had been established for sequencing the human genome. And actually, if we look at the figures for a bacterial genome, Illumina is overkill. You get much more sequence than you actually need to assemble a bacterial genome. 15, even the small instrument, MySec, 15 gigabytes of sequence, whereas the bacterial genome has 1 to 3 million base pairs in its sequence. So running a bacterial genome on the MySec instrument would give us a coverage of 5,000 times, which means that each part of the genome, at least in principle, would have been sequenced 5,000 times, which is much more than you actually need. The solution to this would be, for instance, to sequence more than one genome at a time by so-called multiplexing, where you start with, for instance, what we have done, 12 or 16 or 24 different bacterial genomes. You fragment them, you tag each fragment with a specific oligonucleotide, which will allow you to tell from which organism a given sequence is derived. And you run all these multiplexed genomes in the same experiment, in the same sequence. And we have, in my work, we have used up to 24 times multiplexing, which I write here works fine. We'll see, I mean, it may be that it would have worked better with less than 24, maybe. But anyway, we can solve the too high capacity of an Illumina instrument by running more genomes in each experiment. So Illumina will give you, for bacterial genomes and even for the human genome, more or less the full genome, but it will be present in fragments, in contigs, where each of these green contigs here have been obtained by joining together thousands or millions of reads from the sequencing instrument by looking for overlaps at the end of each sequence. And again, for many purposes, it doesn't really matter if the genome is fragmented, because the fragments anyway will cover at least most of the genome. But again, a fully assembled genome can show us things that a collection of contigs will not tell us. So it's certainly, in most cases, it would be at least prettier and more satisfying to end up with a circular genome like this instead of a lot of long but not connected fragments. So how to get from a collection of contigs to an assembled bacterial genome? We have discussed this already, and again, the simplest solution, although not the only solution, but the simplest solution would be to obtain sequence reads that are long enough to span all possible repetitive elements present in the genome we are interested in. One solution would, of course, be to sort of adapt an Illumina instrument to read longer sequences. As I said, the MySeq instrument would be able to read up to 300 nucleotides in a sequence, but not more than that. And this figure tries to illustrate why there is a limited read length when we sequence clusters of DNA like we start with here. So this is sort of one of these clusters containing up to 500 copies of the same DNA, which we saw on the Illumina substrate. And we have performed the first sequencing reaction here. We have added a yellow nucleotide to all the fragments here. And we identify this by looking at the color and can tell that in this case, we have incorporated a T in the sequence. So we remove the fluorescence, we remove the blocking group from the 3-hydroxyl group of the nucleotide at the end here, and we perform the next reaction. And we will again have signals from almost all of the fragments here, but some of them will not be able to incorporate a nucleotide. Things can have happened during the unblocking process here, or things can have happened in other ways, which makes it impossible for this end nucleotide here to accept a new one. So from this step to this step, we still have a strong signal, but the signal will be a bit lower than it was here. And for each step we perform here, the same thing will happen. So after a number of steps, which for a MySeq instrument would be maybe 300 steps, we will end up with signals in so few of these identical fragments that we no longer can distinguish signal from noise. So at this stage, we have to stop somehow. We can't read further because the signal is not strong enough to allow further reading. And the same goes, if you think of it, actually for all sequencing methods that rely on sequencing a number of identical molecules at the same time. In the Illumina case, in the same cluster on the substrate. I mean, these biochemical reactions are like normal chemical reactions. They will proceed, and if they're efficient, they will go to almost 100%, but not all the way to 100%. So for each step here, we are doomed to lose a part of the signal, and in the end, the signal will be so low that we no longer can read sequence. The solution to this problem is, at least to my simple head, very surprising, because the solution is actually to, instead of sequencing a number of identical molecules, one can sequence a single molecule. And by sequencing a single molecule, you will actually end up with signal to noise ratios which are unaltered for each step here. We start here with one single molecule. We add something at the end, which is yellow and tells us it is a T. We go to the next step, add a new nucleotide, which we can identify as an A, and we continue like this as long as we can. And we can, even after a number of steps here, the signal to noise ratio will be the same as it was in the beginning. And this is because, of course, I mean, as long as the addition of nucleotides here happens, it will continue. And of course, working with a single molecule, things can happen, which means that we no longer get any sequence here. Things are broken here. We never get to this stage. But that doesn't really matter, except that we'll get a shorter sequence than we otherwise would have gotten. But the important thing here is that as long as the reaction proceeds, we will have a signal to noise ratio which is unaltered, which means that if we have a very good system for this, we should in principle be able to read thousands of nucleotides of sequence from a single molecule, provided that we have methods to read signals from single molecules. And that's, of course, quite a challenge. In the Illumina instrument, we had, say, 500 copies of this tagged nucleotide in a given position. If we work with a single molecule, we only have one molecule tag, and we must be able to detect that one in some way. And that, to a simple mind, seems like a very difficult problem, but it actually has been solved in the instrument which I will present now, the instrument from Pacific Biosystems. They have developed a sequencing technique which they call this is a smart sequencing. And in this slide, I have tried to illustrate how it actually works. The instrument, again, is built up of different compartments where sequencing reactions are performed by a polymerase. But in this instrument, these compartments are very small. The dimensions of this well here, which has, as you see, a yellow polymerase at the bottom here, and the DNA, which actually is in the process of being replicated. The dimensions of this container here is smaller than the wavelength of the light which is used to detect the fluorescence. We have what we call a zero-mode waveguide. And for a small compartment like this, this means that the light we use to excite the fluorescent molecules here in order to be able to detect them will not actually enter the compartment at all, but only enter it at the very bottom here. So we get light which can promote fluorescence into the waveguide, but only at the very bottom of the waveguide. And what they have done here is to put a polymerase at the bottom of the waveguide. It's immobilized in the bottom of the waveguide, and this polymerase will be supplied with a number of fluorescent nucleotide analogs, which like the one we see here. And these molecules, which are sort of inside the waveguide here, will not be excited by the incoming light because the light will not reach them. Only nucleotides at the bottom here will be excited. And most of them, I mean, if this molecule suddenly drops down here, it will in microseconds disappear out again because it will diffuse around. Only nucleotides which are bound by the polymerase will be present at the bottom of the waveguide here for a time which is long enough for it to be detected. So here we have a immobilized polymerase. We have a template DNA and a growing chain of complementary DNA. And in this case, the polymerase will incorporate a yellow, which we can say is a T here. It will bind the substrate, and while the substrate is bound to the enzyme, and this is, it will be bound for at least several milliseconds, long enough for it to be detected as a, well, it's a C in this case. So we get a C pulse because the polymerase at this stage has bound a yellow C nucleotide. Then the nucleotide will be incorporated. The fluorescent tag will be removed. I will show you in a little while how this happens. And we will no longer get a signal from this one until the next, in this case, a blue nucleotide is bound by the polymerase because it's a T in the template, so we should have an A in the growing chain. It's bound and immobilized for a time period which is long enough for it to be detected. So the output from this instrument, at least if you look at this level, will be a series of pulses of different fluorescent signals, where the color of the fluorescence will tell us which nucleotide has been immobilized to the polymerase at this stage. This illustrates more or less the same thing, except that this figure shows us that we have a number of these zero mode waveguides on the substrate here. So in each of these black regions here, we have an immobilized polymerase which will synthesize DNA based on the template sequence. And again, if we look at one of these ones, we will the light we use to excite the fluorescence will not reach further than 10 to 30 nanometers into the waveguide. So only molecules at the bottom here will be excited and send out fluorescence. And the substrates they use for the reaction are very clever. You have a nucleotide here with a normal triphosphate at the end, but at the end of the triphosphate, we have a fluorescent group bound. So that when the nucleotide is incorporated in the DNA, pyrophosphate is removed from the end, and the fluorescence will be removed at the same time. So what we incorporate is a normal nucleotide, whereas the tag will be removed together with a pyrophosphate. And again, since this happens on single molecules, the strength of the signal will not diminish as we continue the polymerase reaction. It will either stop completely if something has happened to the polymerase, for instance, or it will continue with the same signal-to-noise ratio as it had originally, which again will make it possible to read very long sequences. So again, this figure shows us the polymerase here. We have a growing chain here. We have an incoming nucleotide, which will be incorporated in this growing chain. And what this again illustrates is that based on the fluorescent signal, we should be able to read the sequence. This slide also shows that one should be able to distinguish modified nucleotides from normal nucleotides based on in this case, the distance between the blue signal here and the next signal, the green T signal, which is coming afterwards. Since we in this case have a red methylated adenosine in the template, the polymerase will spend more time in order to incorporate the T here than it would in the normal case, where we have a normal A, unmodified A in the template position. So not only can we read sequence, but at least in some cases, you can also tell whether we have modified bases in the template, which of course is an additional bonus to this one. The time scale here is that for each of the incorporations here, I mean, in this figure, it takes about 1 second for each incorporation. In real life, they claim that at least tens of nucleotides are incorporated per second and identified in this reaction. And in this way, and with using this instrument, you can read nucleotide sequences that are thousands of nucleotides long. At least up to 25,000 and even more than that, which is sort of on a completely different scale than what old sequencers like I are used to, where we on good days could read 800 nucleotides from a given template. So sequencing single molecules, first of all, makes it possible to continue sequencing, continue the sequencing reactions without losing signal, and also to be able to read extremely long sequences. The main problem with the Parkview instrument is that the accuracy is not very high. I mean, you can imagine, I mean, we are working with a single fluorescent group here, and you may have problems detecting all of the signals here. For instance, this small t here could easily be missed and so on. An error rate of up to maybe 15% is so high that it will cause problems when we try to assemble sequences, larger sequences from the separate fragment sequences that we get from the instrument. But the people at Parkview has gone to large steps to improve the sequence quality, and the solution to this at the present stage is that they start with something like this. This is a purified double-stranded DNA. They ligate special adapters at the end here so that you end up with a molecule which is actually circular once you denature it. And you immobilize, you put this immobilized polymerase in the correct position, and it can read several times the same sequences here. So in this case, it will read both the direct strand and the complementary strand, and it can read each of them several times, as long as the polymerase is able to continue. The polymerase they use, as far as I know, presently in the Parkview instrument is a bacteriophage polymerase called Phi29, which is highly processive, and they also modified it to sort of stabilize it, so that they claim that at least 70,000 reactions can be performed on an average by each polymerase. And since the errors, as I said, we have maybe 15% errors if we do a single read. These errors are randomly more or less distributed along the template, which means that if we read the same sequence once more, whatever errors we have will not be in the same position as in the first run. And if we read it even more times, you can end up with a very accurate sequence. And the accuracy they claim for this hi-fi technology, which is used in the Parkview instruments these days, is 99.9% accuracy, which is more or less the same accuracy as you get from an Illumina instrument. Except that the reads are at least 100 times longer and even more than that. So if you look at the output from one Parkview instrument, it can put out up to 4 million reads in each run. So 4 million templates are sequenced in each run, and the read length can be up to 25,000 nucleotides in the hi-fi experiment, where both strands are read and read several times to give us this high accuracy. So I discussed this only yesterday with some people at the Norwegian Sequencing Center, and they say that the hi-fi reads from the Parkview has completely transformed the way they assemble or sequence genomes. They work a lot with fish genomes, and it's enormously easier to obtain an assembled genome than it was previously, using only short reads from an Illumina instrument. And they recommend also that even medical doctors should look at the Parkview technology, because although short reads can give you a lot of information from the human genome in disease and health, these long reads will add another dimension to it all. There are other ways to sequence single molecules. And you can even sequence them without using polymerases. And the technology I will discuss now is the so-called nanopore sequencing, where the principle is illustrated here. We have a DNA which is to be sequenced. We pass a single strand of this DNA through a pore in the membrane. So this is an either artificial membrane or not even a membrane at all in some technologies that are being developed. We pass the single-stranded DNA through this pore, and we read the sequence by looking at the variation in the ionic current through this pore. So in a very schematic way, as we pass this single strand through the pore, we can read the sequence of the strand by looking at variations in the current here. So A, C, T, G, and so on. And this actually works amazingly enough, I would say. What they do to get it all to work is, among other things, to use a double-stranded DNA and use a gyrase in this position to separate the two strands so that only one of them are passed through the pore here. The purpose of the gyrase, which should be sitting, or is sitting here, the unwinding enzyme, is mainly to sort of put a brake on the passage through here, because if you don't break it, the DNA will pass through the pore so quickly that you can't read sequence. So the unwinding is mainly here to make the speed of to reduce the speed of the DNA strand as it passes through the pore. If we look at the actual instrument, it has some different implementations, but the typical Oxford nanopore instrument is shown here. And if you compare the size of this one to the size of a factory used to sequence part of the human genome, and if you compare it to the Parkview instruments, it's incredibly much smaller. This is simply something looking like, well, I don't, a small hard drive or whatever, which you couple to your PC. You pipette your samples onto some flow cells in this instrument, and the sequence is read based on these fluctuations and transferred directly to your laptop. And again, this is sequencing of a single molecule. So as long as the DNA is passed through the pore, you will be able to read sequence to the extent that you can read it without loss of signal noise ratio, which is sort of one of the secrets behind this. So they have this small Minion instrument, which can be where one can put several Minions into one unit to get a higher capacity for sequencing. They also, at least some years ago, maybe even still trying to develop a smaller version, which can be coupled to your mobile phone so that you can sequence directly using your mobile phone instead of a computer. But anyway, we have several instruments and with different capacities. But the Minion is sort of the core instrument. It will read 512 sequences, so it has 512 of these flow cells where sequencing is read. So the number of sequences we get per run is much, much smaller than we get from, for instance, a Parkview instrument or from an Illumina instrument, where we are talking about billions of reads per experiment. But the read length can be extremely long. Turns out that I think the current record for reading a single molecule of DNA is 4.2 megabases, 4.2 million bases from one molecule, which means that you in principle could sequence 512 different bacterial genomes in one of these Minion instruments. In real life, of course, that would be difficult to achieve, but you can at least, as I said, obtain read lengths which are even a couple of orders of magnitude larger than the read length you get from a Parkview instrument. But the sequence quality is, the accuracy of what you read is not extremely high even for this instrument. An error rate of 15 to 20% is what one would expect, which again is a bit too high to make it really easy to assemble bacterial genomes using this technology alone. But like the Parkview group, they have implemented some variations in the system to improve the accuracy. So they have, this is what they call a 1D instrument, where a single strand is passed through once and read, and with an error rate of maybe up to 20%. In this 2D experiment, they have joined the molecules together at the end here, the direct strand and the complementary strand, so that they first read one strand of the double-stranded DNA and then the other strand. And in that way, they can improve the accuracy considerably. And they have a variation of this, where again, both strands of the double-stranded DNA is read, but using a different solution to this problem. So using technologies like this, they can improve the accuracy up to towards 99%, even for the nanopore instrument. This figure shows us in red here the size distribution of templates fed into the instrument, and in blue the number of bases that are read, the distribution of read lengths from a single experiment. And what this hints at is that once a molecule enters the nanopore, it will be read completely from one end to the other, which I think is quite impressive. And again, read length up to 4.2 million bases. I find the Oxford nanopore technology to be very exciting for several reasons. And one reason is that even in the system they have today, there's little lab work that needs to be done in order to sequence DNA. They have kits which would allow you to prepare the templates even in the jungle, so that you don't really need a lab at all in order to sequence DNA. And I imagine that future developments in this sequencing technology will make sequencing completely independent on a molecular laboratory lab, which means that you finally get some real democratization of sequencing. Anybody can sequence DNA in 10 years, I imagine. No matter what the lack of microbiology or molecular biology experience is. So I would guess that, again, anybody can sequence DNA, and DNA sequencing will be completely liberated from the need for a molecular biology laboratory. So molecular biologists will not be needed, but you probably will still need a handful of bioinformaticians in order to get the full benefit of the sequencing. The read speed from the Minion instrument is 50 to 250 bases per second, which is again quite impressive, but again means that in order to read 4.2 million bases, each experiment has to go on for several hours in order to reach from one end to the other end of the template DNA. So we have two different systems for getting these ultra long reads. We have the Parkview system and the nanopore system. And thanks to these technologies, the human genome has finally been completely sequenced. It happened earlier this year, 20 years after the publication of the draft human genome. We now have the complete sequence, including centromere regions, telomere regions, and you name it. And this was achieved using the long reads from Parkview hi-fi instruments with 99.9% accuracy, and these ultra long reads from Oxford Nanopore, which would be able to span even the most complicated repeat regions you would find in the human genome. And they also used a very weird cell line, which is essentially haploid, to obtain this finally completely completed human genome. So I've already hinted at what I've written here. I mean, the really intriguing part of nanopore sequencing in my head is that you can, in principle at least, imagine reading sequence without any kind of chemical or biochemical reactions. No need of enzymes and so on. What you need to do today is to purify the DNA and sort of put on some adapters which allow this unwinding enzyme, which you need to break the passage of the single strand through the pore. But with future technologies, even these things maybe can be omitted. The instrument presently can sequence both DNA and RNA, which is very interesting, that you can actually look at the transcriptome without having to convert it to cDNA in order to sequence it. And you can identify modified bases, not with a very high precision, but at least to some extent. And people are even trying to develop systems for sequencing proteins using nanopore technology. So this is what I intended to say about the technology. And to sum it up, I mean, we have methods available these days. On the one hand, for getting huge amounts of sequence using Illumina sequencing, really huge amounts of sequence data. And on the other hand, technologies which allow us to read each sequence extremely long, which, as I've hinted at, will to a large extent solve the problem of assembly, and at least when it comes to bacterial genomes, make it possible to assemble the sequences into a continuous circular genome at the end. So now I will switch gears and try to tell you a little bit about why I got involved in sequencing bacterial genomes. So this is the hands-on part of it all, and as I've said, it will not really be hands-on because we are on the net, but at least I will go through some of the problems that have occurred to my group when it comes to sequencing bacterial genomes and what we did to solve those problems. But first, I have to introduce a group of antibacterial peptides called bacteriocetes. This figure here shows us two ways they can be sort of classified according to each other. We don't really need to bother about that, but the bacteriocetes I've been working with belongs to this group here, which is a group of unmodified peptides, which means that they contain the normal amino acids, which we find in proteins anywhere. And the bacteriocetes are genetically encoded, so we have genes for each of these various peptides, unlike some other antibacterial peptides, which are synthesized using huge protein machines that can incorporate amino acids that are completely different from the 20 amino acids we find in proteins. These bacteriocetes are produced probably by most bacteria in one way or another. The ones I have been involved with are produced by lactic acid bacteria, and it seems that they produce them to kill or hurt close relatives, because most of the bacteriocetes we know have a very narrow spectrum when it comes to what kind of organisms they can kill. We have a few examples of broad-spectred bacteriocetes, but the typical bacteriocene will only kill certain groups of bacteria and not have any influence on others. And they are, as I've written here, they are genetically encoded, and they occur together with other genes in the same operon, genes which are responsible for the transport of the bacteriocene out of the cell, and also a protein which will make the host, the one who is producing the bacteriocene, resistant to the bacteriocene itself, an immunity protein. Bacteriocetes have been studied for quite a number of years, and all through these years, it has been vigorously debated how they actually function. One interesting thing with bacteriocetes is that many of them are extremely potent. It means that you only need nanomolar concentrations of a bacteriocene in order to kill a bacteria which is sensitive to the bactericin. So they are extremely potent compared to many other compounds that will kill bacteria. And why they are so potent, at least one explanation for this was obtained by a group at the Norwegian Life Science University close to Oslo, which managed to elucidate how one specific bactericin, lactococcin A, manages to destroy the recipient cell. I should also say that cells which are killed by these bactericins, at least the ones belonging to the group I've been working with, are killed by the formation of pores in the cell membrane, which will destroy the membrane potential and destroy the metabolism of the bacteria, which eventually then will die. What the group at the Life Science University in Norway managed to show was that this lactococcin A functions by interacting with a specific protein complex in the bacterial membrane, the so-called, let me see if I remember it, mannose phosphotransferase system, which is responsible for the transport of mannose and other sugars from the outside to the inside of the cell where they can be used. These lactococcin A molecules will interact with this system and presumably lead to an opening of the transport system so that it remains constitutively open and will cause destruction of the membrane potential. So the explanation for the high potency of this bactericin would then be that one or a couple of molecules are sufficient to destroy this transporter protein in such a way that it remains open all the time. This group at the Life Science University were able to show that this mannose phosphate transferase system was the recipient of the bactericin by doing some immunoprecipitations of the immunity protein, which is the protein that makes the cell that produces the bactericin immune to its own bactericin. So you could show that this mannose phosphotransferase system co-purified, co-precipitated with the immunity protein in immune precipitation studies. And by this gave them a hint to what could be happening, and they showed then in a series of papers after that that mutations in this membrane system would influence the degree of inhibition from the bactericin and so on. So this was a quite important result because it showed at least one possible mechanism for how these bactericins could obtain their extremely high potency. At my university, we also have a group, had a group which was working with bactericins. It was led by one of my colleagues, Jon Nismeyer, which is about the same age as I. So when I started to get close to retirement age, I realized that I would have to join forces with somebody else in order to be able to get money to do research at all. So I joined Jon Nismeyer's bacteriocin group, and our main goal was to identify other of these receptor proteins in bacterial membranes, which could explain the function of bactericins. Lactococcin A was already solved, so we looked at some other bactericins, and we're mainly interested in bactericins which consisted of two peptides, which both had to be present in order to kill the recipient cell. One such two-peptide system is a bacteriocin called lactococcin G, which consists of two peptides, one which is 39 amino acids long and one which is 31 amino acids long. And they are, as shown up here, encoded in an operon in the genome of the producer, where we have these small protein encoding genes here. And we have some transport proteins, the blue ones here, and we have the green immunity protein, which is produced from this gene here. So together with Jonas and Meyer, we tried to sort of repeat what had been done at the Life Science University in Ås by doing immunoprecipitations using lactococcin G, or the immunity protein for lactococcin G, immunoprecipitations to see whether they would pull down any interesting membrane proteins from the host. But the results we got were extremely dirty and not very interesting. We did immunoprecipitations and we identified a large number of lactobacillus proteins which were pulled down together with this biotinylated lactococcin G. And we got absolutely no hints at all which of the membrane proteins in the recipient cell which could be responsible for the action of lactococcin G. And then I got my sort of good idea, because I've been involved with DNA sequencing most of my life, and the chance to sequence a bacterial genome was something I can resist. So I, in one group meeting, suggested that, we have some mutants which have become resistant to the bacteriocin. Why don't we simply sequence their genomes to see what mutation causes them to be resistant to the bacteriocin? If this lactococcin G would function in the same way or use the same mode of action as lactococcin A, it should interact with a membrane protein and open a pore in the membrane, and the resistant mutants would then be mutants in the receptor proteins, which, for instance, caused the lactococcin G molecules not to bind at all or something like that. So that was the general idea behind it. If we can't find it in other ways, we will sequence the mutants to see what makes them resistant to lactococcin G. This figure down here shows an inhibition zone. So this is a lawn of sensitive bacteria. We have put a drop of lactococcin G solution in the middle here. So cells are killed, but you see some white specks here and there, which are bacteria which have become resistant to the bacteriocin by mutations. So these are the ones we picked and tried to sequence. So the reasoning, my reasoning was that if this resistance which the mutants had acquired were due to mutation, which was a likely explanation, sequences from an Illumina instrument would contain all the information we would need to identify the mutation. And since the complete genome of the sensitive strain we used was also known, I sort of imagined that it would be easy to pinpoint the mutations that caused the resistance. But of course, if one takes an objective view at this, it could be other irrelevant mutations in the genomes. There could be sequencing errors. It could be all kinds of noise, which would make it difficult to detect the interesting differences in the genome and separate them from the noise differences. And since we didn't have much money, we didn't want to recruit a group of bioinformaticians to help us with this. So we would try to do it ourselves as best we could. So we grew up the resistant mutants. We isolated DNA from them, and we sent them to the Norwegian Sequencing Center in Oslo. And in this experiment, we used an Illumina high-sec instrument, which at that time could only read 100 base pairs per read. And we multiplexed. We used altogether 12 genomes. 5 of them were from our resistant mutants, and the 7 others were some other group who also wanted to sequence some bacterial genomes. And the result was 20 million reads per genome, each of the reads about 100 base pairs. So more than enough sequence to discover if we had any differences. So we had a lot of sequence from the mutants. The question then was, how can we compare these mutant sequences with the reference genome and discover what actually causes these mutants to be resistant to the bacteriocin? So I spent a lot of time scanning through the literature and hit upon a program called VAL, which other people had used to find a single mutation responsible for bacterial resistance to some antibiotics. So this was more or less exactly what we needed. I mean, VAL would take Illumina sequences, map them to a reference genome, and detect differences between the sequences and the reference genome. So I arranged to have this program installed on a Unix machine cluster at the University of Oslo, and did some trial and error, and ended up in the end with using only half of the reads from the machine. And even with only half the reads, the program would use 24 hours in order to map the reads to the reference genome and detect differences between the reference genome and the mutant DNA. And in addition, I used a program suite called Geneos on an iMac to keep track of what was happening and get nice illustrations of the mapping process and so on. So after we ran our 20 million fragments through, or 10 million fragments through VAL, what we ended up with the output from VAL was something like this. We had hoped for one single difference between our mutant genomes and the reference genome. Instead, we ended up with more than 150 differences between reference and wild type genome. 152 SNPs, single nucleotide polymorphisms, or differences in one single nucleotide. 5 insertions of more or less, well, not very long sequences, but anyway, and 13 deletions compared to the reference genome, which was kind of a letdown for us. But this was when we compared the reference genome to the sequence we had from the unmutated Lactococcus genome. So, which told us that even though it was the same strand as the strain as the one which had been sequenced and put in the databases, there were differences in the sequence. And once I looked into these differences, 160, 70 differences between the sequence we had gotten and the reference genome, I realized that most of them actually are due to errors in the database sequence, because for instance, these insertions and deletions, which we had discovered, were actually able to recover broken reading frames in the reference genome, so that we actually got a repair of genes which had been missing bases already in the sequence. So of these differences, I guess that almost all of them are not real differences between our wild-type genome and the reference wild-type genome, but errors in the sequencing of the database genome. But if we then looked at the mutants, we found the same 170 or so differences, but in addition, we found specific mutations in addition to these, which were common to all mutants. We analyzed 5 mutants, and 2 of them had the same mutation and introduction of a stop codon in a protein called UPPP. And the 3 other mutants had a frame shift mutation in the same gene, leading to termination in position 86. So we had 5 mutations, but there were only 2 separate mutational events. So we had managed to pick copies of the same resistant colonies from several positions. But anyway, we found 2 mutations in a single protein, UPPP, which encodes a protein called undecaprenyl pyrophosphate phosphatase, which is, as we will see in a little while, important for the maintenance of the peptidoglycan layer in the bacterial cell wall. So 2 mutations in the same gene, we felt that we actually needed more mutants, so we rapidly instigated the process of isolating more resistant mutants, and also picked resistant mutants from another sensitive strain, in addition to this strain which we had been looking at the first time. Yeah. And the result we got was very convincing to us and even to the referees. In all the resistant mutants, we found mutations in the same gene, this UPPP gene. Most of them would lead to a truncated polypeptide, and the truncations are seen here. So this is the wild type protein with these ovals representing membrane spanning regions. So this is a membrane protein. And more or less shortened versions of this protein were produced by the mutants. And in addition, 1 mutant had a mutation in the promoter region of the gene. Another had a mutation in the ribosome binding site of the gene, and 1 had a point mutation at the very beginning of the gene. But the other mutants were sort of mutations that led to a truncated protein. So this was really encouraging results, and we continued to work on this protein to show that it really functioned as a receptor or a recipient protein for this bacteriocin, and that binding of the bacteriocin to this protein was responsible for destruction of the bacterial wall. And the thing that really clinched it in the end was that we could show that if we transferred this UPP protein from a sensitive strain to a completely different strain, which sort of in its wild type version was resistant to the bacteriocin, we could get a strain that actually was sensitive. So presence of the intact UPP protein was enough, sufficient to make a cell sensitive to the bacteriocin. So this worked extremely well, we felt. And we immediately threw ourselves at other 2 peptide bacteriocins to find out if they had the same mode of action as this one. Well, this is a figure showing more details about the mutations we have in the UPP protein. The green here are transmembrane regions. The blue ones are outside the cell, and the red ones are inside the cell. So this is a protein that spans the membrane several times. And the mutations are sort of located in different positions here, but it seems that to remove this last transmembrane region is sufficient to destroy the function of this protein as a receptor of lactococcin G. So if we remove this last transmembrane region, the cell will become resistant to lactococcin G. This shows how this protein actually functions. We won't have time to go through this in detail, but this figure shows how the peptidoglycan layer in the cell wall is produced using a lipid called lipid 2 as a carrier for the whole thing. The lipid 2 will then liberate at the end here the peptidoglycan, which have been synthesized, and we get undeca prenyl pyrophosphate here. UPPP, our enzyme, then removes one of these phosphate groups and also flips this lipid over to the other side of the membrane. So from being on the outside of the membrane to the inside of the membrane. This function of UPP as a flipase was only solved a couple of years after we published our results. But it seems that this pyrophosphatase not only removes one phosphate group, but also moves the whole lipid from one leaf of the membrane to the other, where it can continue in this circular way to produce peptidoglycans. We had two peptide bacteriocins, plantaricin EF and JK, and we wanted to see if we could identify the receptor protein as the same way as we had done for lactococcin G. But for these two bacteriocins, we didn't have a database reference genome. It's a genome here and not gene, which means that we would then have to assemble for ourselves the complete genome of the sensitive strain and use that assembly to identify mutations which could make the cell resistant to the bacteriocin. So we picked resistant mutants in the same way and isolated DNA and sequence as before. We also, of course, took the sensitive wild-type strain and sequenced in the same way, except that we used the MiSeq instrument to get longer reads and cheaper sequence because we didn't have that much money. And we did multiplexing up to 24 times, which means that we could run more strains per run, but we will get less sequence per strain. But still, we ended up with 1.5 to 2 million reads per genome and a coverage of about 200 times, which is more than enough at a reasonable cost, I would say. So for the assembly of the genome, I mean, this is what we would have wanted, but this is what we ended up with. A lot of more or less long contigs, which altogether we hoped would cover at least most of the wild-type genome. And again, I would stress to you that the main reason for getting fragments rather than a complete genome is that the genome contains repeats which are longer than the reads that we get from the sequencing instrument. And I've hinted already that there are some problems. I mean, it could be preferred to have a complete circular genome rather than a collection of contigs. And the reasons are, first of all, that the contigs are wrong, at least in the end. Otherwise, they would have been assembled further. So that we will observe differences between the reference genome and the mutant reads that don't reflect actual differences between the genomes, but simply problems or errors in the assembly process. And the contigs will not necessarily cover the entire genome, which means that if we were unlucky, we could miss interesting mutations because they were not part of the contigs that we ended up with. So this is the results for plantaricin JK. Again, 2 peptides which both have to be present in order to kill the cell to be killed. We picked 11 resistant mutants. We assembled the genome ourselves. And for the assembly, we had to try several solutions. The first thing I tried was an assembler called Abyss. But we got more than 500 contigs, and this program we used to map the reads to the reference genome was not able to detect any differences at all. So Abyss was not a solution. Velvet, another assembler, never managed to get less than 1 million contigs, which is a lot of contigs from small bacterial genomes. So we had to stop using, or had to find other alternatives. I tried something which, to my pleasure, would run on a standard Mac, a genome assembler called Spades, but ended up with 737 contigs, and Val would not accept this as a reference genome. So I became desperate, but fortunately, this program suite I had used to keep track of the sequences also had a built-in assembler, which I tried in desperation and managed, after a couple of tricks here, which I won't go into, to get an assembly of 243 contigs that Val actually accepted some reference genome. So we could finally start comparing the resistant mutants to the wild-type sensitive genome. But we ended up, when doing this comparison, rather than having one single difference between a given mutant and the wild type, we had between 15 and 27 differences, of which some could be due to real differences between the genomes, whereas others obviously would be due to errors in the assembly. And when I looked at these differences that we did observe, and looked at where in the contigs they were placed, many of them were placed in contig regions where the coverage was very low, 1 to 3 reads rather than 50, which was the average in this experiment. So these ones I could also conclude that probably were due to defects in the assembly rather than real genetic differences between resistant mutants and sensitive wild type. So I removed these irrelevant differences and ended up with genomes with between 1 and 7 differences compared to the wild type. And these ones I mapped to the contigs and used a gene finder for bacterial genomes called Glimmer to predict genes in each of the contigs, and ended up with, again, 10 of the resistant mutants having a mutation in the same gene, a gene which encodes a member of the APC superfamily of transporters, which are responsible for transporting amino acids and polyamines from the outside to the inside of the cell. So 10 of the 11 mutants, resistant mutants, had mutations in this gene, and they were, many of them, localized in a specific region of the protein or the gene. And the 11th mutant I could find out also had mutations in the same gene as shown here. The problem was that the mutant genome must have been contaminated with a wild type genome, because instead of having 100% of the sequences having a mutation in this position, only about half of them had it. But I felt quite safe in concluding that even the 11th mutant had a mutation in the same gene. And this is how the mutations were placed in the protein. And again, we have protein spanning green regions here, inside the cell in red and outside the cell in blue. And if we look at the model of the protein, or this protein family, it turned out that this is the inside of the cell down here and the outside of the cell up here. And almost all of mutations were present in the intracellular loop between the 6th and the 7th transmembrane region, and some were in this border between the 7th transmembrane region and then loop outside the cell. So again, we felt quite safe in concluding that for lactococcin JK, this membrane protein functioned as a receptor protein, in the sense that this is the protein that would be destroyed, or the function would be destroyed when the bacteriocin interacted with the bacterial cell. So binding of the bacteriocin to this protein would again open the cell membrane so that the transmembrane potential would be destroyed. Which again, I mean, taking into account that we had started without a reference genome, I felt quite pleased with the results for even this protein. And I could actually verify that the mutations I had picked were the correct ones by a couple of months later after publication, getting a Pacific Biosciences assembly of the whole genome. Extremely long sequences, but this was before the hi-fi technology had been invented, so it was sequences with up to around 13% error rate. But anyway, it could be assembled into a single contig, which was 1.6 million bases long. Which, by the way, I realized after a while was not really correct, because I couldn't join the ends together to make a circular genome without getting weird results. So even the Parkview contig was not correct, although it was much better than the 250 contigs we had from the shorter reads. So we could verify all these mutations and get rid of the noise from due to the incorrect assembly of the genome. We also had yet another 2-peptide bacteriocin, plantaricin EF, which we also wanted to find the receptor for. But for this one, we had really problems picking resistant mutants. For the other bacteriocins we have discussed so far, resistant mutants emerged without much effort at all. But for this bacteriocin, we got mutants that were somewhat resistant, 20 to 40 times resistant, which means that we would have to use 20 to 40 times more bacteriocin in order to kill the cells, but not really the complete resistance that we had obtained for the other bacteriocins. And we could use the same sensitive wild-type genome as we had used earlier, so I didn't have to do more assembly. But we ended up with, for the mutants we had, 3 of the 4 mutants had mutations in a gene, which is sort of shown here, a short protein, which is anchored in the membrane, apparently at the end here. So 3 of the 4 mutants had mutations in this gene, and 2 of the mutations were identical, which means that they were not independent. And the mutations led to problems in the protein. The fourth mutant genome, we didn't find any mutations in this gene at all. So to sum up the experiments with plantaricin EF resistance, we had few mutants and none of them very resistant. So we tried to produce more mutants by looking at other sensitive strains and see if we could pick anything from them. And I won't go through the results here because they were, we were actually not able to conclude anything at all from these ones. And my guess is that for this bacteriocin, plantaricin EF, I guess that the membrane protein that it interacts with is essential for the cell, so that if it's mutated, the cell will die anyway, which would, of course, then make it very difficult to pick mutants. So we got a lot of sort of hints when it comes to mutations, but nothing that could explain or nothing that we really could feel convinced about really interacting with the bacteriocin itself. And I tried different ways of assembling the genome and didn't get any new results from that one. So for plantaricin EF, we didn't really get any results. The last one I will describe briefly is again a bacteriocin called plantaricin S, where we in the beginning had managed to get one single resistant mutant from this EL 1403 strain, which was the one where we actually had a database genome, which sort of should simplify or at least reduce the noise somewhat. And what we found for this resistant mutant was one single difference, and C to A mutation in the codon in a gene for a hypothetical protein called YTHA. And this mutation led to a stop codon, so it would lead to a truncated protein. And the truncation would lead to the removal of a cytoplasmatic tail in the protein. So this is the operon of this ITHA gene, and this is a position of the mutation. And the consequences of the mutations, this is a mutation, and it would lead to loss of this C-terminal part of the protein. Which we are not really sure whether it's inside or outside the cell. And ITHA is an interesting protein in many respects because other people have looked at other bacteriocins to try to find proteins that, when mutated, make cells resistant, and ITHA has turned up in several such studies. And we also found that this truncation of this protein led to increased resistance to several bacteriocins when we compared them, and also to resistance towards a couple of normal antibiotics, especially kanamycin and aminoglycoside, hygromycin. No, not that one. This is G418 disulfate, which was 3.5 times more resistant once we had removed this tail. But again, even for this protein, we have reason to suspect that cells get a bit resistant, not because the protein actually functions as the protein which interacts with the bacteriocin, but for other reasons, because even for this bacteriocin, we were not able to get really resistant mutants, but only mutants which were somewhat resistant. So to conclude my experiences with bacterial genomes, The conclusions are that it's actually possible to use out-of-the-box solutions. I mean, programs available on the net and elsewhere to detect and locate mutations in bacterial genomes without having to rely on a lot of bioinformatics expertise. It certainly helps if you have a reference genome available in the databases, but we were, at least for some of our studies, able to assemble genomes that were good enough to be able to detect differences between resistant mutants and sensitive wild types. And the final conclusion with this, I mean, things worked well in those cases where we could pick mutants that were really resistant, but we get less clear results in the cases where resistant is less than 100%. That they are somewhat more, they can take somewhat more bacteriocin, but if you put more of it in, they will eventually die. Which is, I mean, bacteriocins are interesting for many reasons, and one reason is that, I mean, it's kind of counterintuitive to use a peptide to destroy a protein that the owner of the protein actually can do without. I mean, these resistant mutants were quite viable, except that they would no longer be able to produce cell wall in the same efficient way, but using alternative solutions. And they would not be able to transport amino acids in the same efficient way as before. And for the mannose phosphate system, they would no longer be able to transport sugars into the cell as efficient as before. But anyway, it's sort of hints to me that the main reason why bacteria produce these bacteriocins is not really to kill their close relatives, but to tell them that if you are staying in my neighborhood, you should not eat mannose because I will have that, but you can eat whatever else you are able to eat once you have become resistant by destroying your mannose transport system. If I were to repeat this work today, I would make a couple of differences. I would absolutely include Parkview, preferably hi-fi sequences, in order to get complete genomes, or genomes which at least does not consist of hundreds of contigs. And again, the reason why this would be better is that contigs by nature are contain errors at their ends at least, and these errors will make noise when you are trying to detect specific mutations in a mutant genome. And I would use simpler tools for mapping the mutated reads to the reference genome. For instance, this Snippy here, which has, again, for me, the advantage that it can run on a normal desktop computer rather than a Unix system. But I mean, the take-home message here should actually be that bacterial genomes, it's fun to sequence them. It's not that difficult, and the sequences can give you real interesting results. And why interesting results? I mean, we know these days that bacteria on us and in us are much more important for health and disease than we suspected before. For instance, the gut microbiome seems to have vast influences on things like obesity, depression, and things like that, which one wouldn't really expect. So these genomes are important to study. And again, what is even more interesting to me is that if you look at all bacteria on Earth, we know something about 1% of them, whereas 99% of them are completely unknown to us because we have not managed to cultivate them in the lab. But by doing metagenome sequencing, where we isolate DNA from the environment or from the gut, from water, from soil or whatever, and sequence everything we can get out of it, we will get some idea about the diversity of organisms in that environment. And there are several examples that doing metagenome sequences, people have been able to assemble complete genomes of bacteria, which again were completely unknown to us because we have not been able to grow them in the lab. And by having the genomes, we can get some idea at least about their metabolism and the way they live, and the genome may even give us hints about what we would need to add to our cultures in order to grow them in the lab and study them further in the lab. The final message, in biblical terms, if we make a small modification of what God said to Eve and Adam, he said that we should be fruitful and multiply and replenish the earth and sequence it and sequence the fish of the sea and the fowl of the air and every living thing that moves upon the earth. Again, my take-home message is that don't be afraid of attacking genomes because it's not that difficult and you don't need a lot of expertise in order to do it. These are the people I work together with in the bacteriocin studies. Camilla and Bie were PhD students who managed to get their PhD, partly because of the genomes we analyzed, and Jon Nissenmeyer was sort of the man who has worked with bacteriocins his whole career. And these are the people at the Life Science University of Oslo, which also aided us in our works. And some thanks to the Norwegian Sequencing Center and to the guy who installed WALD on the Titan cluster of the University of Oslo and helped me testing it out. That's all I have to say. And I mean, if anybody has the power left to ask questions or comments, I will happily answer them.
Yeah, thank you, Tom. I think, I'll ask for a few questions maybe. But how about this part related to the hands-on? You included everything into one, I guess.
Yeah, I did. Yes, yeah, sure. So this last part was a hands-on part of the whole thing.
Yeah. Okay, so questions from the audience, please.
Could I ask a question, please?
There is one question in the chat, and the question is, in your experience, how many times can you wash the cell of the Oxford nanopore and reuse it?
I must say that I don't have any hands-on experience with the nanopore sequencing. I only sort of admire it from a distance. And I mean, once I get to the stage where I can sequence genomes in my kitchen, I certainly will save up money to buy one of these instruments. I mean, it's $1,000 for this 512 flow cell device, which will give you lots of sequence. So it's not expensive, but expensive enough for me to prefer other solutions. I mean, nanopores are nice, but with the hi-fi modification for the Parkview system, they are in most cases more useful because the sequences you get are more accurate. But if you need really long sequences in order to join together parts of the human genome, then of course the nanopore.
And you can take several.
But I don't know how many times you can reuse a nanopore thing. I'm sorry.
There is a question about the UDP phosphatase mutant.
Yeah.
If the UDP phosphatase mutant confers resistance to bacterial bacteriocin, is still functional.
Yeah, it's not functional, but the cell will so I mean, you destroy the enzyme that normally is responsible for removing one of the two phosphate groups from this undeca-prenyl pyrophosphate. But there are other phosphatases in the cell that can take over this job if the UPP protein has been destroyed. So the cell will still be able to produce its cell wall, probably with a lower efficiency than with an intact UPPP protein, but still able to function. So the protein and the gene is not essential for survival of the cell.
Other questions? Please don't be shy. We still have time. Otherwise, I will invite Professor Wessel Christensen. No more questions at the moment? Hello, Wessel.
Do you guys hear me?
Human genome variation.
Yes. So you know what? First of all, congratulations to Tom to this great talk. I don't know him from that perspective that well. So that was really nice.
Very nice.
Okay. Then shall we start?
Yes, please.
Здравейте на всички! Надявам се да не се разбягахте сега в тая пауза. Не много често ми се случва да си поздравя така аудиторията на български и затова веднага използвам възможността. Много се радвам и благодаря на Антон и на Игор за поканата и възможността да участваме в този курс. Той се получи малко теоретически, може да се каже. Надявахме се да имаме възможност да поговорим също и за вашите проекти и как всъщност можем по-директно да включим практически методите, които използваме, но да се надяваме, че по-нататък и това ще се осъществи. So the title of my presentation, The Human Genome Variation in Health and Disease. I work at the Department of Medical Genetics at Oslo University Hospital. Our department consists of several large sub-departments, the one of research, which I lead. We also host the Norwegian Sequencing Center, so a lot of the sequencing experiments, some of the sequencing experiments that Tom described have been performed in this sequencing center. We have 4 MySeqs, 4 NextSeqs, and 2 NovaSeqs that have extremely high throughput. We reach out for projects both in human and bacterial and in fact mouse, any animal genomes. I will depart from, so do you hear me well?
Very well.
Very well. So I will depart from Tom's presentation in several directions. So first of all, he gave a very deep and profound overview of how the reading of the human genome or any genome works, how sequencing works. I will depart from the bacterial to the human genomes, and I will depart from the sequence as such, and how the message from the sequence is turned into biological activity, and what function this biological activity, and how do we study the function of this biological activity in both health and disease. So the next, my slides don't want to move, really?
Frozen.
Oh, okay. Another way to move it. Okay. Maybe not the arrows, but the mouse. Okay, I can use the mouse. So how do the message, which is coded in the genome, the so-called cookbook, all the recipes, do translate into into a message. So in the human genomes, just as it was already said, it's a lot larger. We have close to 3 billion characters, so 3 billion base pairs that create the human genome. Basically, as all living creatures on Earth, these genomes are created out of 4 letters. It is a 4 letters alphabet. In the case of the human, all the text is recapitulated into 23 chapters. These are the chromosomes. And we have something like 25,000 genes, plus minus estimated, which are in a way the carriers of the meaning. And all this is studied through a chemical reaction, which has been evolved since the 1970s when it was first introduced into higher and higher throughput DNA sequencing. So the reading of the whole human genome will create a big book, a large book. How do we read this book? One could draw a couple of parallels, and of course, assuming that you will be people both from the clinics, taking PhDs in different fields, not exactly genetics, I thought I would start with one very basic description of the terms. And if you do take your PhD in genetics, I hope that anyway, even if I'll be speaking about things you know from before, I may get you to think about these things in a little bit different way, hopefully. So if we compare how information, if we look at genetics as an information science and compare how information is moved along the lines, how the message is transferred in both language, data science, and genetics, one could identify a couple of parallels, of course. Both work with letters. In the case of genetics, there are 4 letters. In every alphabet, there are close to 30. In data, it is a binary code that is sufficient to describe all the variety of signals that one wants to transcend. Letters are organized in syllables or words so that we can pronounce them, or we can compose a message into something, and data message is integrated into processors. And in the genetics, we have something called triplets, or something that codes for the specific amino acids, which is the exchange bit between the genetics and the real in the real biochemical world. Also, in language, these words are composed into sentences, and in data, we work with well-defined programs that have specific functions. And in genetics, these programs could be looked upon as genes that code for specific proteins. Then also sentences telling a story could be combined into a paragraph that takes a mini story inside the big story, which is somewhat comparable to genetic locus on the human chromosome, where we have several genes that could be transcribed together or maybe controlled by a common control locus. We have chapters, which I already said are comparable to the chromosomes. And then we have certain different books that have their own different stories, which can be different cells and organisms. And those all can be combined into libraries, which could be compared to large complex networks, and all into populations and society. So being a geneticist means that one then should be able to read a book as large as this, orienting oneself into what of this is a meaningful sentence. Tom said that not more than 1-2% of all this is actually worth reading. Also, in a book like that, where is my full stop? Where is my capital letter? How do I identify the beginning and the end of a sentence? And what of this has biological meaning? So compared to other thick books, we know War and Peace. That was a book that had close to 1,200 pages and 538 different words. And just as there, we know that there are certain chapters and there are certain ways to kind of process ourselves through a book like this, which is quite large, and the letters are small, and we don't have a clear start and stop of a sentence, and not everything gives meaningful text. And the letters are then chemical compounds, so we need chemical reactions to be able to, in fact, read them. And to be able to understand them, that many as they are, we need to visualize them in some way, and there are multiple methods in which every numerical value in a given biological experiment is given a color, and then we can use these to look for similarities between, in this case, these are patients and different genes. And more of these methods we will be discussing tomorrow. So we know that there is a certain organization in this, that these 3.14 billion nucleotides, they only give rise to 1 to 2% of the words that we use in this word, that we have something between 25,000 genes in those 46 chromosomes, one from mom, one from dad. And then we have close to 42, these give rise to close to 42 millions of proteins in a cell. And a living human has something close to 373 trillion cells. So these are quite a lot of cells. And we also tomorrow, we will be speaking about methods that give us the possibility to, in fact, study tissues now at a single cell by cell basis. We know that, yes, DNA is transcribed to RNA, to a messenger RNA, which is transported to the cytoplasm and synthesized into protein, which is the central dogma of the molecular biology. We have the genome. This is the mass which is inside the nucleus, all the DNA, which is then organized into chromosomes. When stretched, it is available for different proteins and enzymes to come and approach it, like transcription factors, elongation factors that actually do create the mRNA, which is related to protein, which creates the molecular machine in a living cell. It was also quite important, not only for the cell itself and for biology itself, to have this information separated into different chapters. It was also important in the Human Genome Project, because then every chromosome was given to a different lab or different of these transcription of these sequencing factories, which Tom described, which had its goal to sequence their way through their particular chromosome. So, and in this stretch of the DNA, we discriminate between genes. Here they are described in very, very, very schematic and short way. In fact, there is no gene that is that short, the 4 nucleotides. The shortest gene there is 1 microRNA, which is 21 nucleotides long. And the longest gene there is, that's the one of dystrophin, which has 2.3 million such base pairs, as you see here. But in fact, speaking about genes and learning, decomposing everything into genes and studying their function has been, through the history of molecular medicine, been a very important factor to discover different diseases. A gene can be looked just like a sentence, has its own syntaxes. It's not an amorphic mass of letters. It has a capital letter. It has what is it called, object, a subject. So we have something which is the controlling area, the promoter. A gene in the human genome, in contrast to bacteria, is composed of so-called exons and introns, which are further on introns spliced out into a mature mRNA, which is the one that creates the protein itself. So, and then identifying these promoter sequences in this large book is, of course, the factor that tells us where a capital letter is. There, how we know we have a sentence that starts. And something called a poly-A tail, or the end of the gene, is something that tells us where around probably the full stop of this sentence will be. So there has been a huge work, A, to first chemically read all these sequence, the way it was described in the first lecture, but also B, to in fact characterize all these starts and stops and all these exons and introns and all these genes and functions, and what do they in principle do. And this would not have been possible to know at the level of the Human Genome Project if there was not so much work done before on studying simple genes, similar genes. So when we discover in this amorph mass something that looks like something else in nature, then we can say that, oh, maybe this is a kinase or maybe this is a protease. And in that way, there's been a huge amount of work to pile evidence for the functional importance of these proteins that are coded by the genes. And this became finally possible with the Human Genome Project, which also was mentioned earlier, and which was the final full stop of being able to read all the sequence of a human being. In fact, by 2019, there has been in the world basis, in public databases, 1 million fully sequenced human genomes. That seemed to me like a larger number than I expected. And in fact, indeed, genetic technology, genetics is very strongly technology driven. The sequencing of the DNA is getting more and more available and cheaper for every experiment. We know that the first genome that was sequenced in the Human Genome Project took 15 years to sequence, and with today's technology, one can go down to 1 hour. And this, of course, explains how by 2019 we could be able to end up with that many sequenced genomes. In our laboratory of medical genetics, also the number of diagnostics, high throughput sequences analysis is also been growing with the years. Oh, I'm sorry, it's white letters on white background. So this is from 2013 to 2019 every year. So you can see the number of exomes that are sequenced and the number of reanalyses that were necessary. Because the genomes do have coding sequence in 2% of the whole genome, the initial efforts on looking for disease-associated mutations were, of course, in these exomes. But from 2 years ago, we have moved from exome sequencing into full genome sequencing in our diagnostics. And of course, it's also so that in the human genetics, the actually discovery of what is in this non-coded or this 99% of nothing doing so-called junk, what kind of regulatory and other functions are hidden there, has been a huge topic of research. So the question is, of course, in not only in human genetics, but also in nature, how can one get, is the question of diversity. How can one get so many different morphologies, so many different colors, so many different shapes out with only 4 letters? And of course, one of the explanations is that one can use these combinations of 3 nucleotides, which are necessary to attach to each of the essential 20 amino acids. And with 3 nucleotides, if you get a combination of 3, then 4 to the power of 3, you can actually have unique triplets for 64 amino acids. And in fact, you don't need for 64, you need to just 20, because we know that everything, all the proteins are built with these 20 standard amino acids. And what do we use the remaining for? Yes, those who remember from the university, the triplets or the genetic code is degenerated, which means that there are amino acids that answer to more than one such triplet, which also has its buffering function. So in that sense, one can make different sentences. So like I've tried to make some little example here, like if you take like a sentence like Sam enjoys beer, and then if you can have a little bit different reading frame, so you can have same joins, beer, like one like that. This creates diversity. At the same time, this also I will show later, when a mutation occurs or a change occurs, this can actually create a lot of problems, and that could be a disease causing change of the meaning. Then another thing is also Tom said that the human genome is not the largest. So how come we are the best and the most how can we explain our superiority if we only have 25,000 genes or less genes than many other organisms? So this is also quite an interesting thing. How do we, with those genes, with those available genes, create diversity? And another example that I've tried a little kind of mental riddle with that was that if you write here 3 commands, make food, bring coffee, give the newspaper to the cat, those can be combined in different ways to actually create the one make coffee, if this is more urgent, or bring the newspaper, if that's what follows, or give food to the cat, dependent on what the environment is and what the different impulses are that bring the cells to use one or another mode of combination of these units. And the existence of exons and introns exactly gives us the possibility for that. And it's more and more appreciated that a lot of the protein diversity is in fact due to what we call alternative splicing, or the fact that we use different exons and sometimes even introns to make these different sentences out of the same stretch. So this kind of idea of the central dogma that I described at the beginning of one gene, one protein is no longer completely true. And then, of course, in addition to that, so how can we get the words only with 4 letters, and how can we get so many different sentences with the same building blocks? Another question is, of course, if all cells have the same genes and the same genetic material in each and every cell, as we know they have, how come our body ends up with so many different cell types when they only use the same 25,000 genes? And this is, of course, because not everything is digital, not everything is yes or no. The amount also matters, and which genes are expressed where and to what degree also matters. So first of all, we know that there are epigenetic alterations like this described in this central circle here. We know that the promoters of many of these genes can be methylated and turned off, as opposed to demethylated and turned on. And this is a process which is very important, both evolutionary, embryologically, and also in the development of cancer. We also know, as I spoke about, the context or the gene regions or the paragraphs in a text, that we can have areas in which we, in fact, in these small genomes, like the bacterial genomes, this gene regulation is a lot more straightforward. I mean, there is always so that the gene, and that's how we were able to describe the process altogether, that there is a gene which is either expressed or not expressed, and then there is a regulatory area which determines whether this gene will be expressed or not. But in complex genomes, we know that there is something like 3D structure of the nuclei. We know that areas in which the DNA is not compacted into chromatin can be stretched in different ways and can make it possible that different areas get in contact with each other in different ways, in which if we only look in a 2D way, we will not be able to predict. But if we look at how the DNA is actually placed in the nucleus, we can identify those so-called transcriptional factories of genes that are transcribed together. And this may be either because they are in somewhat vicinity, as you see here, they're at least on the same strand of the DNA, but they can also be on completely different chromosomes and still end up in a proximity of a closer transcriptional activity. So this is very fascinating to me, and some of our projects recently are towards revealing those transcriptional factories. So in addition to all that, of course, I kind of sort of gave you a little refreshment from the school books on how you can make one person. But we are 7.9 billion on Earth. And of course, it is important to bear in mind that in this variety, in this number of people, there is a huge amount of variance. In fact, by 2017, we knew about 324 millions of genetic variants from healthy individuals. from sequenced human genomes. And there is, of course, a gradient from identical twins. Maybe some of you do experiments in data sets with identical twins. They're very precious for genetic studies or studies of the environment, where we know that people have the same genetic background, but during life are exposed to different exposures. But also we know that, of course, this variation across the world is large. Here is given only on the background of India, northern India and southern India. And in 2015, then the typical difference between two individuals was estimated to 20 million base pairs. So this is quite a lot. This is 0.6% of the 3.2 billion base pairs that we know. And most of these variants are so-called single nucleotide polymorphisms, or SNPs, that I'll be coming back to, which represent a simple replacement of one base of those to another from individual to individual. Even in Norway, which is completely, or relatively to India at least, quite a homogeneous population genetic-wise, we can identify areas in which people have a closer resemblance to each other than others. And even those areas, if you genotype people and you just look at how close they are, their genomes to each other, you can see that they kind of recapitulate areas which are valleys or other geographically not easily interchangeable areas where certain populations have been kept more or less isolated. So if we look at all this variation, which now I convinced you is in fact more than one, if one doesn't work with that, might expect, we, of course, have some of these variants can be very strongly associated to disease. Those we call monogenic diseases, because we would know that one gene would cause a disease. We have a large variety of diseases that we know that many different genes have a certain component or a certain contribution. We call those polygenic diseases. And then all these move into something that we may call polygenic traits, which doesn't have to be disease. That could be height, that could be color, that could be temperament. And also the idea of the disease gradient is, of course, dissolving down to genotypes with not known phenotype. And those not known phenotype are with not known phenotype until we discover insult or an infection or an environment in which they suddenly can exert their phenotype. So Igor mentioned at the beginning the COVID, like how many of those variants that we now discover could be associated with severe disease were with no known phenotype until now, or had been known with phenotype associated to different diseases. Also, this pyramid I have drawn myself completely without any opinion about what the size of these parts of the pyramid are, and with all these lines which are there just because I know better graphically. In fact, those lines don't exist, and this kind of gradient from any phenotype to a disease phenotype is very blurry. This is particularly important in psychiatric disorders, where phenotypes are indeed not very easy to distinguish. So if you look at this side here, I have given a little presentation of the types of mutations that we can have. We can have so if you have this could be the normal gene, the reference one. We could have a substitution, we can have a G to T, we can have a deletion, we can have the whole dot here is deleted, we can have an insertion, then instead of the word dot, we have the word fat. And all these different changes create different effects on the correctness of the message. In some cases, they may not lead to anything, so these would be the ones with no known phenotype. But in other cases, they may have, in different degree of severity, changed the message. And in this last one, where we actually have a complete reader frame mutation, as we call them, now nothing after that makes any sense anymore, because then we are in a way come out of frame and the words are no longer having any meaning. In addition to these subtle mutations, we also have structural variants, structural variations, which could be like a whole deletion. There could be a deletion of a whole part of the chromosome. It could be a deletion of a whole arm. There could be an inversion, or there could be an amplification. And in this case, we have two different blue areas of this. And we have in cancer, are quite used to see and study them, but with the more and more genomes being sequenced, we now realize that also in normal individuals, without any apparent connection to the disease, there's quite a lot of structural variation. So in fact, according to the 1,000 Genomes Project, a human can have typically between 2,100 and 2,500 such structural variants, of which then close to 1,000 are large deletions, 160 are different copy number variants. And so this poses a big problem for departments like mine of genetics, which is then supposed to counsel parents on what is really disease-causing and what is not disease-causing variant, given that, in fact, the so-called reference or the so-called idea of what is healthy and unhealthy is so dispersed. And in fact, this idea of the transition of the normal to disease in a more philosophical way, what is really disease and who is actually sick, and what is normal, is taken into this book, The Genes, by Siddhartha Mukherjee, who became a famous author, at least for us in cancer research, because he had a different previous book that was called The Emperor of All Maladies, that described cancer and the history of cancer research quite well. So in summary, this variation compared to a reference genome, one average genome contains a lot of variation, close to 4 million single nucleotide variants. So these are these single nucleotides, insertions and deletions, large insertions, a number of duplications. And also for every new meiosis, there is a new de novo mutations of between 80 and 100 mutations, which when we do analysis of trios, we discover in children, but not in their parents. And something close to 250 to 300 loss of function variants. So this all comes to tell us that, in fact, we do not have a common reference in the genome, which could be used, or, we have something, but let me show you how imperfect that is. First of all, the reference genome is not anything but the sequence that came from the Human Genome Project, which was not a sequence of one single person, but was a combination or a hybrid genome of many people, of several people, 5, I think. And on one hand, there is no guarantee that there are no disease-causing variants in the reference genomes. They were not selected to be particularly healthy. But also, just as I showed you, the more and more we sequence, the more and more we see that there is deviation of the normal. And then the question, indeed, of what is disease-causing and what is not disease-causing becomes very pertinent. And I think that without understanding fully the function of those genes, we will not be able to really give the answer to that. So in our department, we do this identification of variants by a method which is in-house called ELA, in which we first subject the reading of the genomes as after the sequencing, and we also have a number of annotations about how, to what degree they're considered interfering with the function and disease causing. So in cancer, we know that in some cells, and this as well, we are now sequencing increasingly number of normal cells, and we actually identify the number of mutations in those normal cells to be much higher than expected from this model, which you can see from the quality of this figure that it is actually quite old. So this is the so-called two-hit Knudsen model, saying that there is a mutation that occurs in some cell somewhere. Somebody may be born with it. Then we speak about hereditary cancer. In which cells seem normal, there is a mutation, but maybe the other allele compensates for the function. And there is there is predisposition, but not really increased proliferation. If a second mutation occurs in this, then the cells begins to proliferate too much more, and then which in a way proliferation itself brings to replication errors and accumulation more and more mutations and occurrence of malignant cells. So there are several questions if we follow this process. Are there individuals that are at higher risk of cancer or environmental assault, and can we identify the factors, the genes that are involved? Can we identify these early genetic changes and in what way they then change the function of the genome? And can this give us a hint of which of these mutations will get propagated further? How can we use them diagnostically? And then further on with the technology giving us the possibility to do this analysis in a genome-wide fashion, the question is, can we then use this analysis and their expression in concert to help to identify those that patients in which surgery alone could be sufficient to treat a disease, and those who may need a little bit more either chemotherapy or what kind of treatment modality. Also, we know that basically in cancer, the growth, the local growth of the cancer is not really the lethal event, it is the metastases that are the lethal event. So how can we both detect and maybe stop the metastatic process in each particular patient? So this, came a little bit in Norwegian. This is the personalized treatment or the personalized medicine aspect of that. So knowing everything I just said about the genes and how they are built and how they function, how can we identify those individuals that will benefit from a specific drug? And how can we stratify a population using prognostic markers into those who need a given type of treatment? So in the sense of the disease-causing mutations, all these different types of mutations that I just described have a little bit different also call for different treatments. So there are a couple of mutations for which we know that they exist and we already know the mechanism, how they work, and they can be so-called actionable targets. So these mutations can be specifically attacked by a given developed drug. Many of the other variants can be used for pharmacogenetics, which will then identify those who will be rapid or slow metabolizers of a given drug. And then, of course, any specific treatment options for polygenic predisposing genes is more and more difficult. So the more diffuse the genetic marker is, of course, the most difficult it is to target it. So if you look at cancer therapy, for instance, this is a little bit busy slide, but I will maybe try to show you to the things that are important. If you look at how many drugs there are out there, for which there is a number of mutations that are identified by next generation sequencing, for which we know that there is an interaction. So that means that if you have this or this or this or this mutation or mutation in any of those either genes or pathways, then a given drug is the right one. These drugs are 90. And if you look at the number of genes for which you know that if there is a mutation in this gene, I think I know what I'm going to use, I know which drug I may be going to use, this is no more than 130 genes. So here you realize that there is a lot more we can do in identifying this must be just the low-hanging fruit. So there must be a lot more possibilities to identify novel genes that may be predicative either of known drugs or of drugs that can be developed as well. And one such factor, in addition to, of course, the point mutations that one can identify by mutation, by sequencing, are genomic instability and larger structural variations. So this is a cartoon which was actually drawn by hand by someone who was trying to describe into this Virchow archives in 1879 abnormalities of a cancer cell or different types of abnormalities of division of cancer cells. And you can see that all these abnormal divisions, either division of the nuclei but not of the cell, or defects of the mitotic spindle, can lead to different chromosomal instability, genomic instability. And this can be studied either by this method called, this is with radioactive isotopes, C34. Then you can visualize chromosomes very well. And if you are a cytogeneticist and you know how they should look like, how long they should be, and mostly you even don't need to be a cytogeneticist to know that you need to have only 2 of a kind. So every time you see 4 chromosomes of one type, like here, you know that you have an abnormality. So there is a number of malignancies that are in fact characterized with a certain karyotypic variation, and this can be used for their diagnosis. Later on, this multi-fish technique was introduced. So here now you can see structural variants, just the ones that I described at the very beginning. But in addition to radioactivity, you can actually use color so that you can see which piece of a chromosome. You can see that this is longer, but you can also see where actually this long piece came from. So you can actually identify translocation. Later on, once this you can do, however, but you see that you don't have very big resolution, right? I mean, you can divide the chromosome in half and half, but you cannot really see in very small pieces what happens. With a technique called loss of heterozygosity, then you can identify so-called allelic imbalance. So you can see that somewhere in this gel, I have something that doesn't look, if I compare blood and tumor. So here you see this is the blood. I have 1, 2, 3, 4 bands, and here suddenly I have a missing band. So I have a missing chromosome. Later on, when the array techniques came, suddenly it was possible to actually walk all these chromosomes with probes, and these probes could be with such density that you could now see really micro deletions and micro amplifications, and you can actually plot them and see. So here, every of these columns is 1 chromosome. So this is from chromosome 1 to the chromosome X and Y, but this is breast cancer, so there's nothing on the Y. So you can see that everything that deviates from the middle line is actually either amplification, red, or deletion. So there is basically no locus across 100 patients. There is no locus on the chromosomes that in a breast cancer cell is really normal. And then later on, with yet another generation of arrays, the so-called SNP arrays, we were able to identify these gains and losses with even higher precision, because a SNP array not only gives you the idea on this chromosome whether there is something more or something less, but it also tells you whether it comes from the mom chromosome or from the dad chromosome, because the SNPs are often heterozygous, so they are markers of where it comes from, the mother chromosome or from the father chromosome. And we see that it's actually not at all by chance which alleles do get amplified or which alleles do get deleted. And I will give you 2 examples for that. So this just exemplifies like a normal cell where we have 2n, and that we can have a deletion or an amplification, or how these abnormal copy number states arise. Now, so the take home of this part is that in the course of tumor development, the genes and the chromosomes undergo massive alterations. These could be amplifications, deletions, or translocations, which can cause fusion genes. The challenge is that there are many, and it is not so obvious which of them are really important. So which are the ones that are drivers and really drive the proliferation, drive the expansion into metastases, and which are just passengers. They just happen along the way. Then, of course, in these genomics underlie the transcriptional profiles, for which I will be speaking a lot more tomorrow. So this means that these abnormal genomes, where we have all these amplifications, they cause abnormal transcriptional regulation in the whole genome of the cancer cells. And we can see how much that can tell us about the cancer cell, and how much of that is actually regulated by epigenetic effects, for which Thomas Fleischer will be talking tomorrow. And so the real challenge is to identify the interplay between the genomics, what is on the DNA level, the epigenomics, these epigenetic switches of each gene, and how do they shape transcriptomics in the resulting tumor biology. So I will give 2 examples from 2 different clinical studies that we have. The one is the study of tumor heterogeneity and the dissemination in breast cancer. So we know that a primary tumor is in fact built up of very heterogeneous population of cells. And Xavier Tecpli will be specifically speaking in his talk about that. How do we identify this heterogeneity? We know that some of these tumor cells, they enter the circulation, and then they could either go to a distant organ and create a metastasis, or they can stay in the bone marrow and remain into something called disseminated tumor cells. In this state, they can stay for many years, up to 20 years in a apparently healthy individual. And then in 20 years, something happens, something gives a signal of these dormant T cells, tumor cells, to proliferate and enter again the circulation and get into a distant metastasis. So we studied these cells by isolating bone marrows from patients, by staining these cells with epithelial markers, EpCAM and similar, by micro-manipulating them, so trying to catch a single cell of those into a tube, isolating DNA from that particular single cell, and performing DNA sequencing, whole genome DNA sequencing in just one cell. With this, we were able to, first of all, compute the copy numbers, just the way I showed, like whether there is amplification or deletion, and identify the spot mutations, the point mutations. Also, for those that we found that there is amplification in this single cell, so remember, you just have template from one cell, we were also able to go with FISH and design the sufficient probes to actually show that what we see amplified at the DNA level is also amplified in the nucleus. So I will give a couple of examples of cells, such disseminated cells that were found in the different patients. So if you look at this part, you see my cursor, right? I was seeing Tom, so I assume that you also see where I'm pointing. Do you?
Yes.
Yes, thank you. So in this lower plot, you see that two lines. If all purple points were the same line and all blue points were on the same line, there would be no tumor cell, everything will be normal. The fact that we have, so this shows that there is a full deletion of chromosome 4, a full deletion of chromosome 6, and so on. So if we look at those in a way that we have the primary tumor, so we have sequenced the primary tumor, which was easy, there was a lot of DNA, but then we have also sequenced one such DTC. In this particular cell case, you can see that we see quite high concordance between what we see in the disseminated tumor cell and the tumor. It's 98.6 concordance. So what is deleted in the one is also deleted in the other. When we do this several times though, so this is how we publish that. When we do that several times though, if we have 6 patients now, and of these 6 patients, we have something like 30 cells together. And from one patient, we actually have 7 different cells that were captured at time of diagnosis and 10 cells that were taken 3 years after diagnosis. What do we see then? Then it is the same plot. At the very top is the primary tumor. So we use this as a sort of a facet, like a reference. And then we align all the DTCs from 1 to 6 or from 1 to 7, how many we have, according to how similar they are to the primary tumor. And here you can see that if you look at all these concentrated circles inside, we don't see exactly the same thing. I mean, if you look at this one where we had most information, the very out one is the primary tumor, the one inside is the lymph node. And then after that are the different mutations. So you can see that in the lymph node here, we have an event, green, which was not in the primary tumor, which is kept in some of the disseminated tumor cells, but not in all. And so then there's not a minute of boredom. One can sit there for hours and watch these and see how they are different or how are they the same. And we see that those in the center, those cells in the center, they're the ones with the fewest events. And some of them are actually not really tumor cells. So we could go back and see how many morphologically classified disseminated tumor cells did really show to be tumor cells and how many not. And it was about half. That's a little bit, you see that like about of the 24 attempted tumor cells, we could only confirm 10 that they were real. The rest were uncertain or were shown to be clearly hematopoietic cells. And it was also interesting that some of these hematopoietic cells had some changes, like here, look at these two red ones, that were not consistent with the tumor at all. So these were some mutational events that happened in these hematopoietic stem cells, which we saw were associated with age, but not with the tumor process. So this gives you an idea how one can use sequencing to actually not only, of course, compare the tumor development, but even reconstruct tumor evolution by looking at the events, both like structural rearrangements, but also like mutations, compare the frequencies in each fraction and in each cell, and then identify which clones appear from which clones. So in this case, where we had both bulk tumor, we could see that there were some mutations like this 107R, 107F, which were very early events in the tumor genesis. We see that the tumors had more or less the same aberrations described here until some time point where they left to the lymph node. And in the lymph node, we see that a number of new aberrations happened in the lymph node. And in fact, we could also see that all the DTCs in these patients, which are these red one at the end, they all come from the lymph node, not from what we saw in the primary tumor. So this gives an example of what one can do in clinical setting with this technology. Also, in another clinical trial, so how many more minutes? Do I have 10 more minutes?
Of course, yes, sure. Very
Yes, is it okay? 10 more minutes just to show you these other results. So this is a clinical trial of breast cancer where the study design was as followed. Postmenopausal patients, all estrogen receptor positive, were randomized into 2 arms. Now here I need to say a little, I'm lacking the intro, I'm sorry, but this is because, of course, this was supposed to be sequencing talk. But because breast cancer comes in 2 varieties, either estrogen receptor positive, this means there is an estrogen receptor on the surface of the cells, or negative. When the case is, and 2/3 of breast cancers are estrogen receptor positive. When the case is estrogen receptor positive, then the estrogen receptor itself can be blocked. It can be blocked by several ways, either directly block the receptor or stop the production of its ligand. And its ligand is estrogen. So here we have a process, 2 drugs. The one is called letrozole, the other exemestane, that do stop the production of estrogen. And this, it happens here in the core of the aromatase enzyme. The aromatase enzyme is supposed to produce estrone from androstenedione. So androstenedione comes, is bound in the catalytic center of the enzyme and is turned into estrone. So if you bind instead of androstenedione something else, like for instance exemestane, in an irreversible way, then even if androstenedione comes, it will not be able to be metabolized to estrone, and then you stop the production of estrogen. Of course, this is only possible in postmenopausal women, but very, very potent and very common treatment. Another way to do that is, instead of binding irreversibly, you could be just a competitive inhibitor. So the other drug, letrozole, does that. It competitively binds. It may be removed, but at a certain dose, it is so efficient that androstenedione cannot come and bind. Now, both these drugs have been shown to have close to 100% efficiency of blocking estrogen synthesis. No matter how you measure it, MALDI-TOF, mass spec, either way, there is no more estrogen. Now, the clinical question here was, when a woman is given letrozole, especially in metastatic setting, one can have quite good response for given time, months, and then suddenly resistance occurs. The same with exemestane. But when this happens, then a clinician changes the drug from letrozole to exemestane, which in principle does the same thing. It does stop the production of estrogen, but it does work for some time. So they kind of buy time with that, but they do not understand why it works, right? So that's why that was the motivation of this trial. We take patients, some start on this, and then they're switched on this, and then we take patients who start this, and then they're switched on this, and then we take a sample in each of those time points. And this sample, we have subjected to whole genome, both DNA sequencing and RNA sequencing. So we had whole exome sequencing, 556 samples, and RNA sequencing, 41 samples. And also, after that, we have gone through a new technique called single-cell sequencing, for which Xavier Tecli is going to talk tomorrow, and specifically about this trial, what kind of cells you find in these tumors. But if you look at the mutations, you see that at every time point, we identify a number of mutations, both at baseline, at 2 months and 4 months treatment. And one particular sample had a large number of mutations. You can see that this has a really catastrophic events in this tumor. And we also see that if you plot, so here are all the mutations which are common to all 3 time points, and above are the mutations that kind of appear also in a given time point of the treatment. We can plot this in terms of these colors, and we can see that for instance, in this sample 3, which had that many mutations, we can see that on the switch of the tumor at time point 2, the switch of the treatment at time point 2, the percentage of the different mutations representing different clones in the tumor changed drastically. And here we have also been able to produce this kind of phylogenetic trees to see how these mutations occur from one common trunk, one common mutational specter, into different specific to the different clones. Also, when we look at the genes, when we see these mutations more and more often, you can see the usual suspects. You can see MAP3 kinase, you can see PIK3CA. These are all mutations which are characteristic for breast cancer. But also, with exactly the method that I showed to you in the previous single-cell study, we can study the cancer evolution, or what kind of new mutations the tumor acquires after each treatment point. And this is by identifying which mutations, we computationally try to ascertain which mutations must occur in the same cells. But this is only computationally, by juxtaposing the frequency of the mutations to the frequency of copy number events. So we can identify both subclonal mutations and clonal mutations. That means that happen in 100% of the cells. And we can identify all these subclones, which at different percentage happen at each time point of the event. And then we can use this to reconstruct which of these clones come from which, and we can try to identify the clones that are most aggressive, B, the clones that get killed by which treatment, so that this can tell us next time we have a patient which kind of mutation specter would be indicative to which kind of treatment. So of course, all this is done computationally by a former postdoc of mine, Peter Malou, who is now in the Crick Institute and who created a lot of these phylogenetic trees for the Tumor Cancer Atlas project. Now we are currently performing an example in single-cell studies. So we want to indeed take the same samples, disseminate cells in this way, and identify for each and every cell of those whether we see the mutations that we predicted are going to be in these clones computationally. So this happens in a machine called the Tapestry technology from Mission Bio, which we now recently acquired. We are now isolating nuclei from these tissues, and these nuclei will be used into this fluidic system in a very similar way to how single-cell RNA sequencing works. And all these cells will be captured, labeled by barcoding reagent mix, amplified for the mutations we already know are there, and sequenced. So then we will know whether these predictions that we had in the previous slide were correct or not. So this is my concluding slide about precision medicine, which is driven by several disruptive technologies. This is our former health minister. Now we have a new government though, so I need to update my slides. But precision diagnostics is going to be a big kind of a field in Norway in the future. This will be based on DNA sequencing, on both data storage and analysis. We have a whole number of projects on machine learning and how artificial intelligence can be used to mine all the sequencing data that we are acquiring. And all this, by identifying these mutations, will lead towards precision treatment. So hopefully we'll be able to know so much about these mutations with such certainty that we can approach them with gene editing, which methodologically is again completely approachable. So, and this with Francis Collins, who was at the very core of the Human Genome Project, where I actually started my presentation, who also says that all this, although scientifically possible, it requires the appropriate policies for implementation. And we are also here in Norway involved in discussions with both stakeholders, legislation, ethical boards to make sure that this implementation is ethically sound. So I thank you for your attention. It is now past. I just spoke 1 hour. I hope I didn't bore you or do over. But if you still have energy, I can take a couple of questions too.
Okay, so thank you. Any questions from the audience?
I actually could just say something. I asked, because we have the nanopore too, and to this previous, I texted our leader of the sequencing center. He says about the nanopore, right? But he said that he won't use it more than once. He said he will get a wash kit and can then be reloaded, and then can reload the flow cell within a limited time, but only once, I think.
Concerning these single gene mutations, we know that the diseases which caused by single gene mutations are has a very different phenotype. And when I worked with primary immune deficiencies again, I had families with the same, even cytogenetically visible deletion, something when in the family one of the parent who had this event, this mutation, I don't know, this event, has one phenotype, and 2 children has 2 different phenotypes, which are absolutely different in severity, but severity is more, emotional definition. But there are 3 absolutely different types which should be genetically very similar. I mean, surrounding and etc. etc. So we talk about these multiple events in developing tumors. The question is, if we multiply this event for possibilities of their not so easily understandable presentation, because in many cases we doesn't know what is the consequences of each molecular event. The question is if we can interpret somehow this amount of data, this huge amount of data. even with artificial intelligence, because we finally should make a conclusion. If it's reasonable, is it reasonable energy to go so deep if at present, of course, for research, yes, but in not in practical, but close to practical application. If it's reasonable to go so deep to evaluation of tumors, if we really, the goal of personalized medicine is so close to us, or it's just still just some preliminary definition of this approach, because finally we come to very toxic and mostly similar drugs to do. Of course, breast cancer, I'm not specialist, but if there are this estrogen receptor, ER positivity, this is one treatment. If it's HER2 positivity, it's another treatment. If everything is negative, so it's very bad. This we know, but there are still three big approaches, and that's it. And we see so many events. Do you think that it will be finally we close to I don't know, maybe there in parallel some research of small molecules which can be synthesized or I don't know found and attached to specific things and it do something in vitro or whatever. Was it clear?
Yes, I agree with your point. I think you actually raised several points. The first point was, are monogenic disease really monogenic and modifying factors? The second approach is how much can personalized medicine help to identify or to identify the right patient to the right treatment in existing drugs? And the third point was, how does that help the development of new drugs, new small molecules, as you said? So to the first point, the same point is completely relevant for any genetic disease, not only immunodeficiency that you mentioned. It is the same with BRCA1, right? We are in the research consortia to study exactly that, the modifiers of the penetrance of BRCA1. How come we have in our data set a woman who's 85 years old and has had no breast cancer and is a BRCA1 carrier? Right? When the chances of developing a disease at the age of 50 are 70%. So this means that even in the most strong single monogenic disease, not to mention all the varieties of Down syndrome, which is a huge debate in Norway. Like, how do we counsel? There is a big Down syndrome children association of the parents who don't want to prohibit testing altogether. So there are many different points of view here. My take is of course, as the more we know, the wiser we should be. So I think that knowing these modifying factors, one can have a BRCA mutation, which means a repair defect, but maybe can never get so estrogenized as to develop breast cancer. And these are different enzymes, different factors. So I think these things are, I wouldn't say they are not worth knowing just because it is difficult, but of course, this means simply that polygenic diseases should be also studied like polygenic diseases with one factor that kind of dominates a little bit on them. Then on the next thing, given that, so for breast cancer, for instance, there's very strong, very well-defined guidelines how to treat it. It's like the London Metro. I mean, if it's estrogen receptor positive, size this and this, give this. If it is this and this, give that. And then it's like lines. So even though this is so strict, often cases, the number of drugs is larger than the number of possibilities to test. So a clinician is still in front of a question of all the things that I have, what shall I give? And in what order? Like, for instance, this sequential trial was going to answer this question. So talking to our clinical collaborators, the oncologists, they say, of course, we try to give the milder drug first so that we don't reduce the quality of life too much, and then try to buy ourselves time with every next one coming. But there are many questions, even inside the existing drugs, that may guide a clinician faster to the correct treatment by knowing these clinical manifestations. So in that sense, I wouldn't say that this is not useful. Of course, it is a simplification because we work with 300 mutations, 500 mutations. What is the mutation panel? But if you know, for instance, for breast cancer, for lung cancer, these EGFR positive mutations, they have quite good responses. For melanoma cancer, immunotherapy has quite good responses. So we have quite good biomarkers in some cases to really make a progress. And for a proportion of the patients, that works. Of course, even in breast cancer, which is 75% very well treated disease and very well tolerated by treatment is very well tolerated by women, there are unfortunately a group of patients that do not get a good treatment option. So there it is still room to do more. When it comes to the last one, the development of new drugs, then everything had the same, but the complexity is even larger, right? Because with existing drugs, at least it's a definite number. It's a large number, but it's a definite number. And with the mutations, they're many, but they're also a definite number. Now, with the treatment strategies and what exactly will become a good treatment and these small molecules, I know about this p53 inhibitor, which Roche had 10 years research on that. And I know that because it was a Bulgarian who was driving this, and at the end it was not pushed forward. They just closed the development on that. So there it's really, I mean, this is beyond me somehow. How this can guide what novel treatments will be developed. This is indeed a complex question, but I think that we can do quite a lot in guiding the right patients to the right existing treatment.
At least find the steps of development of new clones, switching from one type of treatment, some objectivization of switching from one type of treatment to another when you see this changed clonality.
Exactly. Exactly. Especially if you are so lucky that this new clone that grew instead has also a treatment option.
Yes, of course. Like, I don't know, what I know better, leukemia, for example. We know this Philadelphia chromosome and BCR-ABL gene, and we know that there are drugs, thalidomide, old drug, which finally work against this activity of BCR-ABL, but there were mutations in BCR-ABL and new generations of thalidomide-like drugs work against this fusion gene, but mutated, etc., etc. Yes. So this can be the predictor of this at least, and can be studied and defined for each specific patient, right?
Yes, and you know, this system for the single cell works specifically well for blood cancers. So here we can have some good room for collaboration.
Yes,
At least if I don't make it work for breast cancer, you might hope.
By the way, it's described in male as well, breast cancer. I don't remember details, but
Nutlin was this drug I was trying to remember. The p53 inhibitor Nutlin was developed by a Bulgarian guy who worked, and this was really very close to introduction to the clinic, and then still it was not selling. It was not considered to be a selling thing. So.
Okay. Thank you, Vesela. It's endless story because it creates many specific questions, but many philosophical questions. What should be done and how we should approach to when we try to make bridge between science and medicine. I all the time think about this, and sometimes I disappoint. I have no idea. It's not good and not bad, but I just have no idea what in which direction and what's reasonable to do. sometimes just to pray, but still.
Yeah, well, some
in
faith
molecular
you should have.
biology, yes.
Yes.
Mhm,
Can I ask
mhm.
the other of the did that connect in some way to what you're doing? I really wish we could now I really miss that we are not in person.
Yes, this is correct, but you know we stuck a little bit with hematological studies, but maybe it will develop, and then I'm now more involved. That's why I mentioned this in genetic background of COVID infection. And all these molecules which mutated in the pathways of interferon receptor signaling pathway and toll-like receptor, and there are some associations, and it associated also with autoantibodies against the first type of interferons. And there are many interesting things found till now, and that's what I'm doing. It's not hematology and not stem cells at all, but while we are waiting for more laboratory possibilities, I involved in this study and a little bit have some, I don't know, some intellectual gymnastics. I think that we really maybe it was my personal fault when I did not come to in November or in October or in October to Oslo, and online we did not develop something.
But we will. You'll come.
I will come, and I think we can now, for example, today we talk with a young lady who maybe will be researcher. She is medical doctor, but maybe she will involve and find us if we shall make goals to find interesting selection or I don't know, sampled patients, the DNA samples, or I don't know, RNA samples, cDNA samples, and we will analyze them in your laboratory. We'll see. We are open for all these ways of collaboration, which can be interesting both for you and for us.
Absolutely, absolutely. I'm very glad to hear that.
Yes, because there is a range of patients, and we just, I don't know, routinely treat them. And of course, there are many differences and we can go deeper in terms of specific evaluation of patients and in terms, of course, in terms of research, but even in more practical way. Good, will be good.
Very good.
We will dive in blood.
Very good.
Okay.
So tomorrow we will then depart from the DNA world a little bit and then go into mRNA expression, which is the real title of the seminar.
Yes.
More mRNA expression, epigenetic regulation, and what we can read out of analysis of tissue.
Yeah, we are very much looking forward. Thanks
Yes,
for today, both to Tom and Vesela.
Thank
yes,
you.
Thanks a
thank you.
lot.